Hello. This is Chapter 8, DNA Structure and Chromosomal Organization. In this chapter, we will deal with the structure and the molecular aspects of the DNA molecule. Why? Why on earth should we be interested in the structure of DNA? After all, it's just really kind of a sugar. Well, for instance, 2003, not that long ago, in Asia, many thousands of people were struck down with the flu, and this flu spreads around the world and has a 10% death rate. So a lot of people are suffering. We need to know what's going on here. This, of course, was the flu virus, which we know as SARS, and it was a previously unknown virus. We as a human race, combated this by making a DNA vaccine to SARS. And what they did is they took a gene from the virus and they inject that gene into a cell, and that cell transcribes and translates that gene to make a piece of protein. Now that piece of protein then is the same as what would be found in a SARS virus. It's not the whole virus, it's just one little piece of it. So it doesn't make you sick, but your immune system attacks that foreign protein, and of course once it recognizes that little piece of protein, it will recognize the whole virus and it can destroy it. So we have this capability to make a DNA vaccine against pretty much anything now. The question is, which we'll discuss in class, would you take this new vaccine to see if it worked? How much would you risk to advance technology and to advance medicine? Would you put your health on the line? We're moving now into the history of DNA identification and discovery. And it all starts around about the end of the 1800s. And this chap called Frederick Miescher was dealing with white blood cells in pus, essentially the infected goo that is coming out of patients. These white blood cells, he degraded them with an enzyme called pepsin, and he isolated nuclei from these white blood cells. And the stuff inside these nuclei he called nuclein. And when you analyze the nuclein, it contains carbon, nitrogen, oxygen, hydrogen, and phosphorus, all the common atoms that you'd find in organic molecules. So a few years later, this is in the early 1900s, there was a gentleman called Frederick Griffith, and he was a public health worker in London. And London at that time, lots of people were dying of pneumonia. And so Fred Griffith was researching a vaccine for this pneumonia disease. And it's caused by a bacteria called Streptococcus pneumoniae. And there are two types of Streptococcus pneumoniae. There's the S type, which is known as the smooth type. And it's pathogenic because it contains a capsule around the bacteria that protects it from your immune system. And then there's the rough type, the R type, that's non-pathogenic, and that's because it does not have a capsule and the immune system can destroy it. So he was doing some experiments on mice, and he would inject mice with R strains of bacteria and S strains of bacteria. So here's what he did. He took mice and injected them with the harmless R strain, which is the rough strain, and these guys just lived just fine. If you inject a mouse with the S strain, the pathogenic strain, then not surprisingly, this one dies. If you take S cells and you heat kill them, so you heat them up in a cooker, and you destroy those S cells, and then you inject those into a mice, then those guys are okay, because you've killed the bacteria. But this is the experiment over here on the right that was troubling him. If you inject the mouse with live R cells, if you look over here, live R cells, it works just fine. And plus heat-killed S cells, if you look over here, the heat-killed S cells, they live just fine. If you combine the two of them, the mouse died. Now why would that be the case? Because the live R cells are good, the heat kills S cells are good, but together they kill the mouse. So this was a conundrum for Frederick Griffith, and he wasn't sure what was going on. If you then dig into, if you autopsy this dead mouse here, this dead mouse here, if you autopsy this mouse, you will find that there are live S cells in their blood. So it went from heat-killed S cells to becoming live S cells. The live S cells suddenly appeared. He called this process transformation. And the process of transformation was transferring genetic information. And he went looking for the transforming factor. He didn't know what that transforming factor was. We know it's DNA, but he didn't know. And so we started looking for this transforming factor at this point. So we'll move now ahead to 1944, when Avery, McCarty, and MacLeod did some follow-up experiments following Griffith's work. Now essentially what they did is exactly the same experiment, except that they took apart each of the components of the dead S cells and degraded each different part. So Avery separated these S cells into their chemical components. He treated the S cells with either DNase, which is an enzyme that degrades DNA, RNase, which degrades RNA, lipase, which degrades fats, protease, which degrades proteins, not surprisingly, and enzymes that degrade carbohydrates. So he did this one at a time. So he had 5 different experiments, each one with a different degradative enzyme. And his final conclusion was that DNA contains the genetic information. And that was because the only experiment that worked, in other words, the only experiment where the mouse survived, was when there was DNase was digested. So up here, where it says, when the DNA was digested, transformation did not occur. And the mouse lives. So the mouse was happy. Moving on from Avery's experiment to about 1950s, and the next experiment was by Hershey and Chase. And Hershey and Chase finally proved that it's DNA and not protein that's genetic material. Well, you would have thought that Avery would have done that to start with, right? But it turns out that when Avery published his results, nobody believed him. And we'll ask that question in class. Why on earth do you think nobody believed Avery? And it takes another 10 years or so for Hershey and Chase to actually do that, prove that DNA is the transforming material. So here is what Hershey and Chase did. They're dealing with bacteriophages. A bacteriophage is a virus. Bacterio stands for bacteria, phage means eating. So essentially these are organisms that eat bacteria. And this is how they do it. This is the phage. This thing's on the outside of the bacterial. This is the bacteria here. And when the phage joins to the outside of the bacteria, it injects its DNA into the cell. That DNA is then transcribed and translated by the cellular machinery to make a whole bunch of new viral particles. You can see virus heads here, virus tails and legs and all kinds of things. And eventually that bacteria gets so full of virus that it breaks open and these new viruses are blown out and they go on to infect adjacent cells. So here are the experiments that Hershey and Chase did. There were 2 experiments in parallel, this one on the top and this experiment on the bottom. The only difference between these 2 experiments is that they radio-labeled the protein coat, in other words the actual structure of this virus, with a radioactive isotope of sulfur called S35. The parallel experiment, they radio-labeled the DNA inside the virus with a hot radioactive isotope of phosphorus called P32. The same procedure occurred in both places. In other words, the virus infects the cell, injects the DNA into the cell, and the DNA goes on to make new viruses. Well, when you're dealing with the radioactive protein coat, we find that the radiation is on the outside of the cell. And when you agitate this experiment, and what they actually did is they put it in a blender and turned the blender on high, and it knocked all the viruses off of the cell. So here's the blender moment right here. All the radiation stays out of the cells because the radiation is in the virus. But with the DNA experiment, the radiation goes into the cell. Here it is, in here. And when you knock off the viruses from the cell with the blender, the radiation stays inside the cell. So we have here radioactive DNA, and we find it inside the cell. And this is the conclusion here, that it must be that DNA is the transforming material. So here's the slide that basically summarizes that. It's a few years later, in 1953, so only a very short time later, that probably the two most famous people in molecular biology, Watson and Crick, constructed a model of DNA structure that utilized all this information from lots of other people. This person over here, this is Watson, and this is Crick, and they utilized information from two other people as well, Maurice Wilkins and Rosalind Franklin, who is over here. You can see here. And they actually deduced the structure of DNA. And here is one of the critical pieces of information that they used. This was one of the X-ray photographs that was taken of a crystal of DNA by Rosalind Franklin. And Rosalind Franklin was one of the few people who could actually do this. So her results were absolutely critical to this. So this is what we find as a basic nucleotide structure of DNA. It contains all these different molecules. There's three major parts to a nucleotide. There's this part here, and this is the sugar. This is the ribose. And a ribose is just a 5-carbon sugar. There's a carbon here, 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 and then this one up the top. So it's 5 carbons. A ribose means 5 carbons. This version of ribose down here on the bottom is the same, except it's called, instead of being called deoxyribose, it's just regular ribose. The difference being, look at these oxygens. This unit here is just a hydrogen. This unit here has oxygen and hydrogen. So this top one is missing an oxygen, right? If it's missing an oxygen, it must be called deoxyribose, for no oxygen. This version of ribose, deoxyribose, is what's used in DNA. This version on the bottom, the ribose with the oxygen, is what's been used in RNA. So we have deoxyribose nucleic acid and ribose nucleic acid. So that's one of the components of the nucleotide, either deoxyribose or ribose, depending on which you're going to be. There's another group over here. This is a phosphate molecule, just a regular PO4, nothing unusual about that one. The third major component of a nucleotide is one of these molecules here. These are known as bases. These guys are bases. And they're divided into two types of bases. There's the ones with the 2 ring structures here, adenine and guanine, and the ones with the single ring structures, the thiamine and cytosine and uracil. Uracil is only in RNA. Uracil replaces thiamine in DNA. So what we're saying here is, of course, is it's A, C, G, and T, and that's the DNA, and A, C, G, and U in RNA. These molecules are called, they're split into 2 different types. These double ring structures here, they are called purines, P-U-R-I-N-E-S, and these 3 ring structures over here are called pyrimidines, Pyrimidines.